All right, ladies and gentlemen, let's go ahead and begin discussing the male reproductive system. And really the best way to go ahead and discuss about it is just to discuss it from the perspective of sperm, where it's developed, how it's developed, as well as the things that it passes by in order to leave the male system, as well as anything that will be accompanying along with it. So here's an image of the very basic testis. The scrotum has been removed, the flesh has been removed from it, and this is what you can see. What that is that this testis, this gonad, the male gonad, is actually attached to the male body via spermatic cord. The spermatic cord has quite a few things within it. The ductus vas deferens, which will be the path that the sperm will be taking. It is a tube. We got the cremaster muscle, which it will adjust the positioning of the testis. We will discuss that later on. We'll be talking about the testicular artery. Of course, any type of tissue needs some type of nutrients, as well as the passageways for those nutrients and waste products to pass back into the body. It is also supplemented by an autonomic nerve, which plays a major role in maintaining male reproductive viability, we'll say. Now, here's a Wordy slide. You'll get this Wordy slide back to you here in next go around. You'll have it down in your notes. You can find it in DNA's extras. This is basically what it's saying, is that we have all that information about the testis, the spermatic cord. All this stuff is contained within the scrotum, which does divide the testis into two different compartments, which is useful if you consider if one testis becomes infected, you don't want the second one to become easily infected, so that scrotal septum is there to help keep that immunological boundary. So let's go ahead and dive into a little bit more greater detail into the male gonad, the testis, and you'll see that it has a lot of different tubes, tons and tons of different tubes. The one that we're going to focus on in this discussion here is the seminiferous tubule. This is where the sperm forms. This is where they go from a little blob of a cell to that of the characteristic sperm. What this is called, what that process is called spermatogenesis. Spermatogenesis. I did mention that it turns out like a little glob type of thing, a little glob circular cell, the generic picture that they're kind of showing here. It will go ahead, after mitosis, one of those cells will be sacrificed and be put through the process of becoming a primary spermatocyte. The primary spermatocyte goes on to the secondary spermatocyte, which they achieve. The difference between those two is the stages of meiosis, or meiosis, depending on how you like to followed by the spermatocytes arriving to become spermatids via meiosis 2, and then the spermatids becoming more and more characteristic of a sperm, which is called spermatozoa. At this point, the sperm aren't completely viable and mature. They do have to incubate and mature in a different location, which we will discuss later on. What is interesting is before mitosis, these cells don't really require any type of barrier and doesn't have any selective permeability. But after they go into the primary spermatocyte stage, these Sertoli cells, these sustenticular cells, are there to create a barrier, not only for an immunological barrier, but as well as a nutrient barrier, only allowing certain select stuff to get in. You will see over here that this lays down quite nicely the process in which you will find the primary spermatocytes, secondary spermatocytes, etc., etc. You can see that the ones that are closer to that tunica, that outside basement membrane, are still in that circular form, but as you get closer into that lumen, or that opening of the tube, you'll see those tails starting to droop out. So, Wordy slide, let's move on. The spermatozoa, this is the characteristic sperm that everyone thinks about or draws about when someone says sperm. You'll notice that it has a head and a tail. You'll also notice something really interesting. A lot of the cellular structures that you expect in a cell are lacking. You don't see a rough endoplasmic reticulum. You don't see a smooth endoplasmic reticulum. You don't see a Golgi apparatus. All they really are composed of is a bunch, tons and tons of mitochondria at the base of the head of that sperm. It's that, remember that mitochondria are the powerhouse of the cell, and if they are going to be driven forward, which the tail only allows you to drive forward, the sperm to go forward, it has to have that energy to do so, and mitochondria are the best, most efficient way to derive energy from sugar. You'll also see in the head that it has two different parts, a nucleus, which makes sense. You want the genetic information to be passed along, so a nucleus better be included, as well as you have an acrosome, which will help in the invasion of the egg, and as well as the placement of that nucleus into the female egg, or the female oocyte. Or ovum is another word for it. Here's another slide, wordy slide that you'll have. One last bit of detail that slide is trying to say is that the optimum temperature to grow and mature sperm is not the body temperature. It is not 37 degrees Celsius. It's not 38.6 degrees Fahrenheit. It's actually 35 degrees Celsius. If it were to get any hotter or colder, above or below 35 degrees Celsius, you will have sperm that are inefficient and unable to grow. That's why too tight of a brief can be a problem. It can cause difficult time in reproductive success. So what happens is there's several muscles and several mechanisms that are here to help cool down those sperm and keep them at that 35 degrees Celsius. First off, you have the cremaster muscle, which is in fact an extension of the internal oblique muscle, which if it's too cold outside, the cremaster muscle is going to go ahead and help pull up those testes up closer to the body. You also have the dartos muscle, which is basically almost a lining, I would say, to the scrotum, which is there to also hug and pull those testes close when it's cold. If it gets hot, both the cremaster muscle and the dartos muscle will relax and allow those testes to get further away from the body. We don't want 37 degrees Celsius temperatures near the testes unless they're there to warm them. Another problem is that the artery blood, the blood that's feeding that testis, is 37 degrees Celsius as well. We don't want that sperm to be 37 degrees Celsius. We want them to be 35, dang it. So what we're going to do is we're going to take all that cool blood that's just been hanging out, that are going out through the veins, and we're dispersing it over those arteries. Those arteries are going to be cooled down by the blood that has already been cooled down. So it's a way to make certain that blood that's arriving to the testes have cooled down at least some. So with that, we are going to be, the next slide that we'll be using will be discussing the hormonal control of testicular function. And I will see you all shortly.